This is TWIS. This Week in Science, episode number 1072, recorded on Wednesday, August 19th, 2026. Did science find it yet? Hey everyone, I'm Dr. Kiki, and tonight on the show, we're looking for stuff that will fill your head with cheese, cancer, and cannibalism. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer! As scientists peer into the depths of the universe, find fast stars and evidence for glue balls, questions keep coming about the future of science. Who will be able to do science? Who will pay for science? Who will own science? Why do we worry about these questions? Why do we fret about an endeavor that has historically been closed to so many? Well, if science is not free, then neither are we. So ponder this information in This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about all the science we decided to bring to the show this week. Because we have some good stuff for you. It's been a decent week, you know, lots of stuff going on. Can't rest for a minute these days, but we've got some cancer vaccine success I want to talk about. There are some superpowered supercentenarians that are out there. And I have some cheesy news in addition to how we might actually be able to think ourselves well, the impact of being a sugar baby, and how you move. What did you bring, Blair?
I brought, oh gosh. Stand by. I brought holotypes and I brought sexual cannibalism.
So holotypes, I am going to guess that not everybody knows what they are.
No, we're going to talk about it.
And so everyone went, holo what? And then you said sexual cannibalism and everyone went, okay.
Yeah.
So
yeah, which is a throwback. I feel like I have not talking. Wow, this is going to be an interesting show. I've not talked about sexual cannibalism in a long time. I feel like that was my go-to for the first few years I was on this show. But
very much so.
It's coming back today. So
oh,
nice
it's back
throwback.
in vogue.
Yeah.
It's kind of chic. That should be our meme for the week. Sexual cannibalism. It's kind of chic.
Yeah.
Yeah. Jumping on the meme wagon, you guys. All right. Well, we are here to bring you this show full of science this week. And since you're here, I want to remind you that you can subscribe to TWIS any place that podcasts are found. Look for This Week in Science. Look for that orangey starburst logo and hit subscribe or share it with your friends. Additionally, we are on YouTube, Facebook, and Twitch every week that we are live at 8 p.m. Pacific time-ish on Wednesday evenings. And then finally, if you have questions about the show, want to know information about stories that we talk about, head over to twis.org where our show notes live. And other than that, well, oh yeah, that's also where our Patreon is. You can click on the Patreon link and get into our Patreon community, or also link to our Zazzle store where you can find some of our cool merchandise that's out there. But now it's time for the science. Mm-hmm. Are you ready for this?
Bring it to me.
All right. Bring it down. I know a lot of locations have been talking about this already this week. It is the big story for the week, even though it really hasn't been published yet. This is early.
That's always good.
These are early clinical trial results that have been announced by Merck and Moderna. Now, you might recognize the name of the company, Moderna. And when you think Moderna, what do you think, Blair?
COVID vaccine.
Yes. This has nothing to do with a COVID vaccine. It's all about cancer vaccine.
Okay.
Yeah. So even before the pandemic came, people were working on the idea of using mRNA to allow for personalized vaccines for many different ailments, cancer being one of them. One of the reasons that we end up with cancers is because we have individual mutations that are hard for the body to overcome. And in the case that our immune system can't do it by itself, the idea is that maybe we could help it along. And so these vaccines, and we did report, I think, earlier on the phase 1 and phase 2 trials, this is now the phase 3 trial results. They still need to be analyzed for sure by independent experts, not just the people at Merck and Moderna. But the vaccine that they're using is called Intismeran, and it's given in combination with another immunotherapy drug that's called Keytruda. And the two of them together reduce the amount of time that, well, it increases, I was going to say reduced. It reduces cancer, risk of the disease. It reduces the risk of cancer spreading to other organs, and it increases the time that patients are cancer-free. So if you're going in and you're undergoing a tumor removal or even chemotherapy, this vaccine, once they're using it, the way that it works is they take a little chunk of your tumor, so a biopsy of the tumor. They do genetic analysis of the tumor specifically to see what's gone wrong, where the mutations are. And so then a very specific mRNA tag can be created, and it will work the same way basically that the mRNA vaccine does, where for COVID, where it's basically holding the piece of bad stuff like a flag in its hand, goes into the cell and says, attack this.
Get rid of this.
Yeah, and your body creates a whole bunch of antibodies to attack and get rid of that thing that's bad. And so this is exciting because we know that one of the issues with cancer, in addition to the fact that it's like an eternal cell type, right? It just keeps dividing. They also mutate and continue to mutate and divide because the normal checks and balances have been turned off. And so they accumulate mutations that are very specific to an individual. So you're specific, even though you have maybe you have a liver tumor or a kidney tumor or whatever. And okay, there's this general type of liver tumor or kidney tumor, but you have very specific single point mutations that other people do not. And so because of this will allow the power of the vaccine to be much greater for individuals. And so what they were saying is in combination with the immunotherapy drug, it boosts the immune system. It also increases the attack rate on very specific cell types that hold these particular mutations, and it's helping to keep people cancer-free. And this is skin cancer we're talking about. We're not talking about for this particular cancer trial, for this particular treatment. It is specifically for skin cancer, but it's just a very exciting development because we've been talking about these being on the horizon for so many years. You know, we were talking about mRNA stuff for a long time, and then COVID happened and everything got exciting and woo, yay, this mRNA vaccine stuff is working. How do we get to do it so fast? Oh, because there are decades of work leading up to where we are right now, and thank goodness we have this. And my big question now is, knowing how people have been primed to accept or reject mRNA therapies, I am wondering if in a cancer situation, a doctor, and also not just mRNA, but vaccines. So the name vaccine, which would be priming your immune system to fight something off.
Yeah.
You have cancer. I can give you a vaccine that will help your cancer go away. What is going to happen, Blair?
So, I mean, I have a lot of thoughts. I feel like, so, first of all, people's whatever value you want to put on it, theories on certain scientific
ideas,
accuracies. I don't really know what to say.
Ideas.
People's crazy thoughts about vaccines and how they could be negatively whatever. Anyway, a lot of those things I feel like disappear when it suits those individuals. And so I do think that no matter what your, let's say, beliefs about vaccines are, if you have cancer and you are going to die and you're offered a vaccine that might save your life, I think more people than not are going to take it. So, but I think the other thing I wanted to ask you about is I think the mRNA piece of this is the less interesting part to me. The more interesting part to me is that you said they're going to sample the cancerous cells and make what is basically bespoke medicine, right? And I think, in my opinion, this is the big kind of new revolution, like the next renaissance in medicine is bespoke medicine, right? This is where we're going to start to see massive shifts in life expectancy, massive improvements in medical care that we could never even fathom before is in bespoke medicine. This is the thing that I'm really excited about. Longtime listeners of the show know that I want to live forever, or at least to 150 would be great. And the only way we're going to get there is bespoke medicine. I truly believe that. The only way is to sequence somebody's genome, sequence problematic cells, be able to attack it with precision so that you can have high efficacy and speed and accuracy, right? Yeah. So all of those things together are going to make current potential medical intervention so much more effective. And I love the combination of these kind of two ideas together because they make perfect sense. mRNA is dealing with genetic code. So it's very easy to go, okay, this is your specific cancer cell. Let's go ahead and flag your specific cancer cell. Yep. And I love that. It sounds very exciting to me.
It's very exciting. And in addition, there can also be, banks of prepared mRNA vaccines, right, that are genotyped to certain subgroups, right? So within a certain kind of cancer, melanoma, skin cancer, you're going to have multiple subtypes. So which branch of the cell has gone wrong and started having the crazy mutations, right? So, we can create and clinically prove that these banked ones work. And so you can use overlapped vaccinations to be able to get there faster. But it's just all about being able to genotype the biopsy and genotype the individual tumor, which is
awesome.
It's so exciting. And they're not saying, it's going to get rid of it completely. And they don't know because this is early. This is phase 3. What they've really been doing is going past just proof of concept and getting actually now into let's look at dosage and let's see how this works. And so they're still only at an early level of 1,000 people. But this looks like a really promising trial. And should the independent analysts think that it is great, then it'll go forward to phase 4. And in phase 4, that's when we really see things move more quickly. And so, and a lot of most trials halt between phase 2 and phase 3. Phase 3 to phase 4 is really
exciting.
It's very exciting. Yeah.
That's awesome.
It's something that could work. It's so cool. Yeah. So good news, everybody. Personalized treatments, bespoke medicine. You too can be biopsied and treated. And maybe there was something recently related to aging, and they were saying that based on telomeres and cell divisions and other things, that most of the cells of the body can go on for a really long time. So our kidneys or our liver are almost or liver especially, it's like an almost eternal cell type.
MVP, that
Cell
liver.
types. Yeah. It's the heart, the lungs, and the brain that are the limiting factors. And so, it's really interesting to think of when we talk about telomeres, you think of yourself as this like homogenous thing, you are not milk in a milk carton. You are a very exceptional puzzle. And so you have a really heterogeneous makeup. And so it's our heart and our mind, our heart and our brain that are going to keep us to maybe a maximum human lifespan of 150 to 190 years so far. Yeah. But that's if you do everything right and medicine works. And I'll talk about it in a couple of minutes. You have the stuff to be a supercentenarian.
Oh.
Yeah.
Okay. Don't give me bad news, Kiki. I can't take it.
I'm not going to give you, your doctor's the one who's going to give you the bad news, not me.
Okay,
Come on.
Okay. All right. Do I want to know? I don't know. Anyway.
Yes, you do. What you got?
Hey, what's a holotype?
And I know you like Star Trek, and you're not talking about a holodeck.
I'm not talking about a holodeck. You know what a holotype is.
I do, but why don't you tell us?
Okay, well, I like to think about it like the breed standard in the AKC for a dog, right? So it's like
that's a good analogy.
It's basically like, okay, here's a prehensile-tailed porcupine. This is the prehensile-tailed porcupine that we measure all other porcupines to determine, yes, this is a prehensile-tailed porcupine. So yeah, you're basically like setting up the species standard. This is
we're gonna go with U.S. Science gold standards now.
Yeah, exactly. So it's kind of like this is how big they are. This is what their skin covering looks like. This is what colors they come in. This is how much they weigh. This is how big their teeth are and what kinds of teeth they have. And the reason a holotype is important is to determine speciation. So like, is this specimen that I collected a prehensile-tailed porcupine, as you're looking at on the screen right now? Or is it something else? And B, before you can do genetic testing, you can develop relationships between individuals based on phenotypic similarities. So you can say, like, this is definitely a porcupine. It has quills, like this prehensile-tailed porcupine, but it also has hair. It doesn't have a prehensile tail. Okay, so it's probably related to this, but it is definitely not the same species. So basically, a species is discovered, you develop a holotype, you say this is like the platonic ideal of this species, and then you can measure all other species against it, or specimens.
And this is different from the haplotype, which is genetic.
Right, yes.
Yeah, so it's characteristics versus genetics.
Yes, the phenotypic stuff, yes.
Phenotypic, yeah.
So, why am I talking about holotypes? Well, a study analyzed 1,116 mammal species discovered between 1990 and 2025, so recent mammal discoveries. 95% of the species were native to what we call the global south, which is south of the equator. Latin America, Africa, Asia, the Caribbean, and 60% of the species holotypes are housed primarily in Europe and North America.
I was going to say not the global south.
Yeah.
So we've got biodiversity mostly in the global south.
Yes.
Where it's all housed or stored in the global north.
Yes. Which you can't be that surprised. Like, think about the British Museum, right? I'm not surprised. This is how we do. This is colonialism, right? So
as much as we love our museums, the Smithsonian, like all our museums are like this, yeah.
Yeah. Conversely, only 22% of the species native to the north have holotypes stored outside their country of origin. Not even whether it's in the north or the south, just outside the country of origin, only 22%. And even then, they're stored in institutions with similar geopolitical domains, and that includes things like Australia and New Zealand. So, like, colonial. More than 90% of all mammal holotypes stored in the global north are collected in a different country. In the global south, by contrast, less than 8% of holotypes are deposited in countries other than where they were collected.
Less than 8.
Yeah.
Wow.
So this is higher for charismatic groups, less for things like fish, but it's still a problem. 30% of species described after 2000 have holotypes outside of their country of origin. Reptiles, 50%. The mammal figure is so high. The problem...
I bet birds are really high too.
Yeah, they didn't talk about birds. Always.
But I would
the
imagine,
birds.
yeah.
Yeah.
I mean, migration, they fly up here. They're ours too. No,
Uh-huh. Yeah, right. Depends. Some things migrate, some things don't. But yeah, so this is a symptom of what is termed as parachute science, which is when, you know, scientists come in, they study, they collect, they go out. That's it. They kind of just parachute in, grab a specimen, and scoot. And so why this is a problem is, remember what holotypes are used for. When you are describing what is potentially a new species, plant or animal, you have to have access to the holotype so that you can compare and you can decide whether this is a new discovery or not. That means that if I collect a new South American porcupine and I am native to South America, and I'm like, yes, I think this is a new species, I have to get on a plane with my specimen and go to the place that is holding this holotype to do that comparative study.
So I'm just curious. I mean, I know, having done biology, morphology, these studies, they're the colonial aspect and where things are housed. It's a big deal, and it needs to be addressed, and it's being very slowly addressed, right? But there are going to be infrastructural issues in addressing all this, right? But
yeah, of course.
With modern technology,
yeah.
Why do you have to go anywhere? Why can't we do 3D scanning and put it all online and just do comparisons with
you
people?
Certainly could. Yes.
You're like, hey, put yours online. I'll put mine online, and we can be friends.
Well, you certainly could, but you would have to have a relationship with that other organization. You'd have to have the abilities to have that communication and do the comparative study overseas,
and you have to have all the digital assets.
Yes,
you have
exactly.
to have the technology to digitize, and
exactly. And that's one of the things they talk about in the study too, is genomic analysis techniques that are sometimes used that one institution will have and another institution will not. And so basically just scattering these different specimens causes a lot of kind of logistical questions, aside from just like the ickiness of it too, which exists, right? There's an emotional ickiness, because it does feel very similar to like the pyramids in the British Museum, or the Sphinx, the mini Sphinxes in the British Museum, the mummies in the British Museum. Like, why are they there, right? Like, why can't they be in a museum in the place that they're from, right? And so there's a few different ways that you can kind of attack this problem. There's a few different measures with different success rates and kind of costs and results. So one of them is governance. So that means they want scientific journals to adopt mandatory specimen deposit plans to the current editorial policies so that they can make data and codes available. So this is similar to what you were just talking about, Kiki, like making the data available so that you don't have to pay to go there or to retrieve the data in some way. The next one is infrastructure. So basically that means that you want to create better infrastructure in the locations where these animals are found. Because that's the other piece, is that like the institutions that are taking these animals, these specimens, are usually very well funded. They have very fancy facilities. They have like temperature, climate, and humidity control where they're holding these things. And so most of those are in the global north, quote unquote. So you need to improve the infrastructure in the areas where you are parachuting in and taking specimens and leaving. Right.
This seems like such a place for philanthropies to step up and step in, and whether it is in concert with existing colonial museums, who have the know-how and the ability, but like to do a knowledge transfer, a data transfer, to create infrastructure that actually allows for the transfer of these, the basic assets, and a trusted transfer of assets. I mean, there's just there seems like there should be so many conversations happening to create infrastructure so that, your country's at war right now, we'll take care of your let's get all of the good stuff out and save it, and we'll give it back. There's going to be a record of everything. There's so many things, so many ways that we really should be doing things better. Oh, you don't have infrastructure yet? Well, let's help you do it. Do you want infrastructure, or do you want us to hold it for you? Like,
well, it seems pretty basic to me that if you are an institution that wants to send researchers to a place where there are not state-of-the-art facilities, then you need to have a satellite facility there. Or you need to provide assets to a local university or a scientific center that you can partner with then. Like,
not just you fly there, take things away, and take them back to your museum. The whole parachute science idea is, that's abhorrent. We can't do it anymore.
And then of course the last one is access, improving the access of researchers from the countries where these animals are native to being able to get to those. So providing scholarships to fly researchers out to the holotype if it is not housed in the country of origin. Basically removing barriers to access. But ultimately I was I'm not surprised, but also bothered by this information.
I get that.
I feel like there's a lot of that lately. Not surprised, but bothered, right? Because there's
feel
not a
like
clear
that's me every
solution.
day.
That like you can't just be like, send them all back, repatriate all the holotypes. You can't do that because they don't have a place to go yet. A lot of them, they don't have the right facilities, and it's like logistically, it's a whole mess. Although I will say, like, I don't understand why you need. I understand why. Okay, so hold on.
Too many thoughts in one statement.
I wish you didn't need a singular holotype. I understand why you need a singular holotype. It is for statistical analysis. If you are measuring similarities and differences, and you have a different baseline for one measurement versus another measurement, your data is all messed up. So I understand why statistically you have to have one. I wish it wasn't that way, because it would be great if you were like, there's the North American holotype, and there's the European holotype, and there's the South American holotype, and there's the African holotype, all of the same animal. And so they're all similar. They all have the things that this animal has. Why can't you just use that one? And yes, it's math. Math is the reason.
It's because of math.
Yeah, of course. Which we love math, but sometimes it gets in the way.
It doesn't get in the way. It just creates a short-term barrier that as humans we must creatively overcome.
Yeah.
And we're creative, so we can do that. And I think that's the big thing. You're talking about logistics and everything. It's like, anybody who comes up with reasons why not is just being a gatekeeper, is just, just blocking the path, because what we need now are people who are figuring out solutions to so many things. Of course, there are going to be, oh, we can't do that because, but I want yes and everywhere. Yes and therefore, yes, please. This would be a good place to start. I mean, it would be pretty cool to be able to live for another 100 years and see some of these changes take place. You know, right? So having been here for a good portion, a little bit, half a century, it would be like you want to live to 150, Blair? Like, if you were my age right now, you'd still be looking at another 100 years. So that's a lot of time to be able to solve these logistical problems and to be able to do this. So how do we get there? How do we get to be the people who live to 150, right?
Yes. Tell me.
Yeah. Okay. So study just out in Cell Reports has been making the news related to supercentenarians. What are supercentenarians? Well, supercentenarians are people who live more than 100 years. So and it's not even just centenarians, they have inched it upwards. And so now it's people who live more than 110 years.
Whoa.
So they're
That's
not just,
a lot of people.
So 100 years is a centenarian, but there's people going more. So supercentenarian, and there are people living to 116, 119, 126, I think is like getting like the numbers keep going up, although they are still controversial because of record
errors, right? Yeah.
Because of record keeping 120 years ago. Who was paying?
Yeah, that's the whole blue
actually
zone
recorded
problem.
Yeah, who recorded that birth certificate? I don't know, but I mean, I know lots of people who started counting backwards in their age when they reached 50 or 60. And so maybe they went backwards and then went forwards because they forgot. And so maybe they're even older than that. Who knows?
Oh my God.
Regardless, these provably over 110-year-old individuals, researchers have been trying to study them and figure out, what is it? Is it that you do exercise every day? It's your social? Is it the whiskey you drink? What happens in your life? These researchers published in Cell Reports, CD4 CTLs in supercentenarians. Say that 3 times fast. CD4 cells. Do you recognize what a CD4 cell is, Blair? No, CD4 cells are immune cells. They're cytotoxic T lymphocytes. T cells are the attack cells in our bodies. And so and T cells are the things that attack cancer as well. So CD4 CTLs have been studied in disease context, but not in healthy aging so much. And so these researchers took blood from supercentenarians, looked at their T cells, and they discovered that CD4 CTLs, which are these cytotoxic T lymphocytes, so these immune cells that are, they attack other cells, and they clean things up within the body. They discovered that they start to expand, get to be more of them around the age of 100, and they don't get exhausted. Whereas in the majority of people who are not aging to be supercentenarians, you do not have an expansion of these cells.
So their immune system's better?
Yes. And so that is what is being suggested here. And so they're suggesting that these CD4 T cells, they have really great cloning where they're able to just make copies of themselves over and over again. And cloning is different than normal division, so it doesn't rip up the telomeres as much. And so you end up with, the clones are healthy, everything's good. And so these CD4 helper cells, They can turn themselves into the cytotoxic T lymphocytes. The T lymphocytes clone themselves, and then they can, from the clones, turn into whatever's necessary to attack cancer in any part of the body. And so the issue is, we know that as you get older, your chance of dying from a cancer-related illness increases. Cancer increases as you age because of the number of cell divisions and the greater opportunity for mutations to occur, and for increased mutations within your immune system to be able to suppress the ability of your immune system to actually defend your body against cancer or other diseases. And so what they're saying here is that these supercentenarians have an immune system that stays active and it does not slow down. And instead of being inhibited, it actually expands as you age. And so it is there to address the ills of aging. So we have the question, Long Koenig is asking, how big can their sample be, right? It's not a huge sample, that's for sure. So there are more and more centenarians and supercentenarians in the world. However, they're not able to get a ton of things. And so they've got T cells in blood from 8 donors in their 70s to 90s, 10 centenarians, and 10 supercentenarians. So, their starting sample is very low. But because they're able to compare the genetic aspects of each of these cells, they're able to at least give a general understanding of the differences. And these are the differences that popped out. But we need more supercentenarians to give us cells.
Well, but also, how we're always talking about how like young blood is very good for you. Now I'm like, is super old blood good for you?
But it's for the same reason or for a similar reason, right? So young blood has the markers that refresh the immune system, that refresh metabolism to allow it to maintain at a biologically younger age. And so that's why that works. But this is kind of, you're on the right track there. They're not showing themselves as having an aging immune system in the way that everyone else has aging immune systems.
Yeah. I'm wondering if you can harness that and give that to people who are of middle age.
And so there is the question, right? As we're talking about personalized vaccines, what does a vaccine do? It helps your immune system. So
give me those CD4s.
Right. So instead of maybe having the really healthy clonal population of these CD4 CTL cells, maybe instead what you have is we have treatments or therapies that allow very specific cell populations to be harnessed or created within the body to allow, that address the very specific needs of aging. Yeah.
So this is about like why people live so long and how we can leverage that, but also there's potential medicine in here for just everyday middle-aged humans also.
Potentially. And so if we're able to understand exactly what's going on in those cells and why they're different, maybe it could help everybody. But the researchers here, they're saying this isn't a breakthrough, saving our lives, whatever. No, it's just a very interesting data point that could lead to discoveries in the future, right? So it could lead to specific targets based on what your genotype is, how your immune system works at different ages as you're aging. But anyway, Why do some people have an expansion of these cells as they age? I mean, obviously they need it.
Well, there's also there's the cause and effect of it too, right? Like,
Yeah.
is that what's keeping them alive? Or is that something that you just see in people that age?
And that's another point. Yes. Yep. I mean, those people don't have cancer. When they die, it's usually not from cancer. It is just because they died. So
yeah,
there's a lot going on there.
Hmm.
Yeah. Aging. It's a whole thing.
What about aged cheese?
I love this story. Because really, I love cheese.
Cheese is excellent.
Yeah. So this study came out of the University of Reading, and these researchers in the Food Microbial Sciences Unit were looking at bacteria in cheese. So we've reported on a few studies where we're like, oh yeah, meat's bad for you, dairy's bad for you, but it looks like some cheeses and ice cream might be good for you. And like, there's all these weird results where we're like, what does this really mean? And we have no idea, really. It's just, large data sets of people who ate certain foods and did not have health problems, right? Or did have health problems. But anyway, they looked at biochemical and microbial changes that happened in local cheeses. They took these local cheeses and they were like, we like this. You like the cheese. How does the bacteria affect your gut? So it doesn't just impact the flavor and aromas of the cheese itself. Apparently, the fats and proteins, because of the way they're broken down in the gut, it makes cheese really good at delivering bacteria to your gut. So that instead of the bacteria being killed and broken down in your stomach, because of the fats and other things that are protecting them, they actually can make it into your gut to have benefit down lower in your digestive system. Cheese rinds, if you eat cheese rinds, not wax, the wax necessarily, but the cheese rind, white mold penicillium candidum, it puts rind on soft cheeses like brie and others. It produces chitin, which is a dietary fiber that's a prebiotic.
That's great.
Of course, like myself and other people who are lactose intolerant may understand, mature cheeses that have been aged over longer periods of time have less lactose. So there's less of that sugar milk in there as well. And so the bacteria being involved in that were able to help people eat more cheese. And and those bacteria then are more prevalent in the gut of people who have eaten the cheese, which can help to, over a long term, minimize that lactose intolerance to a degree. So like eating yogurt, eating cheeses that are mature and have the bacteria that break down lactose, that may actually benefit the gut of people who are lactose intolerant and cannot break down lactose on their own.
Nice.
Yeah. So anyway, I thought this study was really fun.
Cheese is good for you. Got it.
Cheese is good for you. Yeah, there are other cheeses that have bacteria that produce propionic acid, which is associated with anti-inflammatory properties. And some people have said eating cheese can help with appetite regulation. Propionic acid is one of the signalers to your brain and gut to stop eating.
Oh,
and
that's
so
interesting.
so the pathway can actually be involved in reducing inflammation and helping to regulate your appetite. So the French were on to something.
Cheese diet. I see.
We are on the cheese diet now, Blair.
Yes.
That's where we're going.
Yum.
Lactose intolerance be damned.
You just gotta buy your lactase in bulk. And
That's
you're all
right.
set.
Full cheese ahead.
Yes. Yes.
Oh my God. Okay, moving on up to that's it for the start of the show. So it is time for us to take a very quick break, which is very quick because really it's just me saying thank you for being here for This Week in Science. We're so glad that you are here. It's just wonderful to see you in the chat room, to see you wherever your comments are, and to know that you're listening. If you enjoy the show, please send your friends to TWIS. Make sure that they're subscribed. Help us grow on all the channels where we're sending this little show out. We've been doing it for a while, but all of your support really helps keep things going. If you are interested in supporting the show on an ongoing basis, head over to twist.org, click on the Patreon link, and become a supporter. $10 and more, and I will thank you by name at the end of the show, but any amount helps. There's even a link on the website for PayPal, so if you don't want to get involved long-term Patreon kind of stuff, just click on the PayPal link and it'll send us a donation, and that is so appreciated. I mean, every little bit helps. There's also a Zazzle link on our website. The Zazzle link will take you to our store where you can find wonderful merchandise. So many Blair's Animal Corner items that are just incredible. And I really hope that you are looking at them for your holiday gifts this year. You better be. No, I'm kidding. I'm not going to tell you what to do. I mean, ask you. Thank you for your support. We can't do it without you. You are what keep us going and keep us unbiased by any commercial influence. So anyway, really, it's all about you. Now we're going to come back to that time of the show we know and love as Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, milliped, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I see what you did. You made a little corner. That's extra cute. Hey, you know how it is. You're a male redback spider, relative of the black widow.
Oh, yeah.
You see a nice, sexy female redback, and you're like,
Auga.
That's the mother of my future baby spiders, my little spiderlings. And I'm like, I know what I'm going to do. I'm going to somersault directly so that my abdomen is underneath that female's fangs while we're mating.
What?
Which means it'll be all the easier for her to eat me when we're done.
Oh my gosh. Why would you
do that? Yeah. Well, there's theories that this will allow them to mate longer for some reason. I guess because, like, while she's eating, I don't know. But it allows them to mate a little bit longer and therefore father more offspring. So even if they get eaten, of course, that means they were evolutionarily successful because they have fathered the most offspring.
That's right. You can go into the void knowing that if you fathered offspring, biologically, you are successful.
You won. Yeah, exactly. Now, we've known about this. We've discussed it before. But this particular study is not about the somersault into death.
It's not.
That the redbacks perform at the fangs of their lover. Oh, but it is when a male decides to do it or not do it when he is hybridized with another spider, and what that might mean for genetics and evolution. Okay, so let me set the scene. So aside from this behavior that exists, some redback spiders who do this behavior have also exhibited abdominal narrowing over evolutionary time, which appears to make it more difficult for the female to bite them during mating, which doesn't keep them from getting eaten.
Oh, but it surely helps.
It delays death long enough for the male to do a second mating event.
How
with
good
that
is the
female.
female? How good is the female redback spider's vision close up? Are they like cats where they can't see in front of their nose?
I don't know. Probably not because they have 8 eyes,
Yeah.
but yeah, so they somehow are able to do a second mating event with that female before they get eaten because they have this abdominal narrowing. Now, this is all in New Zealand. A relative of the redback, the katapo spider, does not do the self-sacrifice. The females do not eat the males. They have no cannibalistic mating behavior. But these two species can mate and produce hybrids in the wild.
What does that do?
Yes, exactly. And this is only
Oh.
possible with katapo females and redback males, since the redback females will just eat the males. They won't mate with them at all. Just like,
Okay.
no, absolutely not. So,
So it has to be the katapo female
because
and a
she won't eat
redback.
the male. Yes. So these are
So
the only successful pairings that allow for hybrids.
what are the genetics of these spiders and what
Yes.
behavior goes
Yes.
forward? Blair.
Yes, yes. What's crazy is those male redback spiders, of course, do the somersault, and they get into the position of like, go ahead, eat me, even with the female katapo females.
That's the male
Yes.
behavior.
Yes. And so
Okay.
that means that the somersault and the cannibalistic behavior have genetic basis because they're doing it regardless of the new stimuli of a different female. They're just like, this is the only way to be.
Does the
This is what we do.
katapo female? Did you say that the katapo female eats the males too, or no?
No, they do not. So that's why this is so crazy that they do that. So
Okay,
they think
got
they're like, all right, we think this is genetic origins. We do not think this is behavioral. We think it's a genetic thing. Okay. So in previous studies, there have been kind of genetic investigations on mating traits, and usually there's a small number of genes or linked gene clusters, and when you do hybrid studies you can kind of see how those genes get selected or not and what influence they have on their behavior in mating. So they found that first generation hybrid males did not somersault, so it's most likely a recessive trait. So then, in order to figure out the genetic basis for these behaviors, they did a back-crossed hybrid. So they did females with males from either hybrid females with males from either species. Then they filmed mating trials with 104 male offspring to determine whether each male did a somersault, developed abdominal narrowing, or both, because they also assumed that the abdominal narrowing was related because it was a mating tactic. So they were like, these have to be similar genes or similar gene loci or like something related. According to their analysis, the mating somersault is consistent with inheritance from a single gene region on the X chromosome. This is way more simple of an explanation than they thought because a somersault is a complex coordinated behavior. It's not just like...
It's a whole process. It's not like
the abdominal narrowing where it's just like, this is a physiological change. Like, no, this is a hardwiring of an entire behavior.
Okay, so we have genes that have led to certain reflexes, right?
Mhm.
So we have genetic basis for innate reflexes. And so, things like hitting your knee with the hammer to see whether you have that reflex. Your tendon has that reflex there in the tendon. It's not even really a nervous thing, right? It's within the tendon.
Right, but this is so crazy because this is like visual stimuli. There's a female, or it could be, it could be hormonal, who knows, but there's some sort of stimuli related
I'm
to
going to
an
do
external force.
a flying through the air maneuver that puts me on my back under her fangs.
Yes. They also found, contrary to what they expected, that the somersault and the abdominal narrowing not inherited together. They were all over the map. About half of the back-cross males had only one of the two traits. The traits are in fact genetically separate. And unlike the somersault behavior, abdominal narrowing did not fit a symbol X-linked or single gene pattern. It's much more complex, which you would expect the exact opposite. Like, this is just a phenotypic change. This should be simple. But that's the complex one. The somersault is the simple one.
Yeah. The somersault, and it's not simple.
Yeah.
That's a multi-step process to make the legs move in a certain way to create this movement. I mean, that is a complex neural behavior. How does one gene, and spiders do not have a large brain, right? They've got a little tiny ganglia, right?
Yeah, they just have like a ball of nerve cells.
Ball of nerve cells. So there's one gene that's going jump across all the nerve cells because of a certain visual stimulus.
So that is contrary to expectation based on what we're talking about. However, the fact that these two species are close enough related, have similar enough genomes that they can hybridize and have offspring, they're true hybrids,
but
that means that evolutionarily they are very closely related and they do not share these behaviors. So it had to happen fast and simply in a short number of mutations.
Yep.
They think that they only diverged about 100,000 years ago. So evolutionarily speaking, that's pretty recent.
So are they still found in overlapping regions? Could this even go further to create a hybridized subpopulation that exists between two separate populations?
Well,
so are they? So this is because they're not sexually separated, which is the definition of a species, right?
Right. So this
sexual
is where you can get into the
separation
really
versus
messy question of like, are these actually two distinct species, or is this one species with different phenotypes?
Yes. Yeah.
And behavioral mating strategies. Like,
yes,
it's.
I mean, the fact that they're together often enough and mating and creating hybrids often enough that researchers noticed it suggests that there's a real blending happening, and they are not sexually, that they are not behaviorally separated.
Yeah.
That males and females are still going, yep, you work.
So, so this is also what's crazy is that
this is so fascinating. Gosh.
Okay, so X chromosomes are thought to evolve faster because of their smaller effective population size in this particular, like spider. But they, for some reason, and I don't understand why, I'd have to look into this further, they think that the sacrificial phenotype was lost. That's the base behavior, whether or not there's a somersault involved, but the sacrifice is the base behavior that then was evolved out in the katapo spiders.
Because the female stopped eating them.
Right.
If the female's not going to eat the male, why does the male need to sacrifice anymore?
Exactly, yes. And so that appears to be what happened 100,000 years ago, is that the female katapo spider stopped eating its mates.
So maybe there always was that. And you know how we talk about beta males, but there's beta females. What about just different behavioral traits that they just got to be big enough populations that we're like, yeah, they're not the same. But you have a katapo male who's like, I'll flip for you.
Yeah. I mean, what is a species? We talk about this enough. Like, I will bring it up one more time. Like, what is a species?
One of my favorite questions.
It's complicated and there's no clear answer. And in this case, if you can have hybridization and you can back hybridize and you can have sexually viable offspring over and over and over through hybridization processes, then you most likely have one species. So honestly, that's the part of the study that I find the most interesting, is that I am stuck to wonder, like, is this really two distinct species, or is it two distinct genotypes within one species that results in phenotypic and behavioral differences? But I don't know. I don't know.
I love this. I love that somebody did this. I want to know more about these. I mean, this is just, these questions get at the basis of what drives speciation. What that question, what is a species? What are we talking about?
Yep.
That's right, guys. Make sure you keep that somersault trait. Might keep you from being eaten.
That's right. Wait, no, it'll facilitate you being eaten, actually.
Oh, yeah.
Yeah.
Yeah. Then no, we won't go any further.
No. I'll give you a quick 2-sentence story to exit out of the animal corner, which is, hey,
right.
you know, carcinization, how everything underwater is turning into crabs.
Yeah. Everybody's so excited about that. Yeah,
that's
it's all
like
crabs.
That's the dramatization, but in reality, there's like 5 different types of animals that have all developed to look like crabs in the ocean. It's because it's a very effective body plan. Well, what about seahorsification? There are these things called sea muppets. Are you familiar with this?
Oh my God.
Sea muppets have been found.
Sea muppets?
Deep muppets, yeah, like Kermit the Frog, have been found at very deep depths.
Deep depths.
Yeah, and so they haven't really been observed by live humans, but they have several I think there are 3 species currently that have been described, and they were all kind of assumed to be related to seahorses because they look very seahorsey. But some CT scanning of these specimens, because they don't have a lot of specimens, they can't cut them open, has revealed their body plan. I can't even really say bone structure. They're like exoskeletal body plan, and it is not a seahorse. It is very different from what we consider seahorses. So even though they look a lot like a seahorse with Miss Piggy for a face, they're not seahorses, but they look like seahorses. So it is a very effective body plan, which seems crazy because seahorses are crazy looking. I don't understand why they're effective at all. They're not like hydrodynamic. They're clunky. They can't move very fast. They're not very agile. They have to grab onto like seaweed and just hold on for dear life. They seem very silly, but apparently extremely effective body plan in the ocean because it keeps popping up.
And I love that, though, historically, everyone's like, it's seahorses. Everybody's a hippocampus.
That looks like a seahorse. It looks like a bad cookie cutter, like a sugar cookie of a seahorse that you got from like Goodwill or something. It's not like quite right.
These poor little guys.
They can't hear you, Kiki. They're dead.
These are dead seahorses, but they're not seahorses.
No.
Yeah.
They're sea muppets.
But I love it. This is, I mean, not everything is a seahorse that looks like a seahorse. Yeah. Just like not everything that kind of looks like a crab is a crab. So
they say like the similar thing you see with mammals is the beaver otter nutria shape.
Yes.
Everything becomes like this aquatic, hairy, weaselly thing.
ROUS.
Yeah, exactly. Yeah.
I don't know. I'm a fan of this.
Body plans that work. They keep showing up. Convergent evolution. That's the whole thing.
Not everything has to be a crab. Sometimes they want to be a seahorse.
Yeah.
And I think that's important to remember.
If you can show me a seahorse crab, you got me.
How would that even work?
I don't know.
Okay, that's
send me your drawings.
That's something for your other podcast.
Yes. No. Perfect.
Yeah, we need Tony Steele in here drawing us pictures of the seahorse crab, the crabby horse.
And then also a crab seahorse. Yeah, those are 2 different things. There's the seahorse crab and the crab seahorse.
Totally. You know. Oh my gosh. Okay, I love it. Not everything is crabs. Seahorses are coming around too. Okay, so I've been thinking about this study this week because I might have gotten stung by a yellow jacket. And I think this is very interesting because we've also had questions about whether or not you can focus on healing yourself and actually heal yourself, right? There are all these cults of thought But there has been through the years, many, many moments where people talk about meditation or you talk about this or that
Yeah.
and how people heal themselves more quickly than other people do. And sometimes people say it's because they thought about, positive thoughts about healing in whatever was injured or whatever needed work. Study published August 17th in Nature Human Behavior. It's titled Voluntary Attention Regulates Acute Immune Responses in Humans. So they had 3 pre-registered within subjects experiments using skin inflammation as a model system. I have skin inflammation, you guys. I really think yellow jackets I don't like them anymore, and I don't care if they are pollinators. I'm going to take action on the nest that is outside my front door under the maple tree.
You can get somebody to relocate it if you really wanted to be nice about it.
No.
You're out for revenge now. I understand.
Oh, they stung me. All I was doing was watering my plants. I mean,
my
it didn't have
goodness.
hydration, apparently. Yellow jackets. My hands, like I have a big hockey mitt and I have like my normal sized hand. It's not a happy time. I'm on antihistamines and all sorts of things. But maybe I'm also on positive thought because according to this study, they found that directing attention toward bodily sensations compared to attentional distraction. So actually attending to sensations. So this is similar to meditation. Breathe in, breathe out. So how does it feel when you breathe in? How does it feel when you breathe out? Thinking about and attending to the sensation. Oh, there's inflammation here. I am attending to that inflammation. Being aware of it and attending to it in a mindful way produced better immune responses with different temporal dynamics. And I think that is like one of the most exciting aspects of this is that there's a temporal aspect of you putting your attention to the feeling of something in your body and your body responding accordingly. And so they had people, sitting in a chair. They were using EEG to be able to manipulate the state. They did a skin prick. There was, allergy stuff that made their skin unhappy. And so they were looking at the time of how the body responded based on whether or not people were paying attention to it or whether they were not paying attention to it, the histamine response. So basically, there's a real measurable histamine response that can be looked at based on your injuries. So first you have, oh, my body's upset. And then the histamine response is that's the itchy part. And so that's why antihistamines work or topical things that get rid of the histamines. It's part of the healing process as uncomfortable as it is. But if you are paying attention to your site of injury, I'm not saying you because this is still a very small study, it changes the timeliness of the response so that your body or the bodies of the people involved in this study responded more quickly and more accurately to the skin offense, the prick,
so
and the
paying
feeling.
Attention to the injury made your body's response to it faster. But that doesn't mean that's not necessarily a better thing. That's not necessarily a good thing, right? Because like you're actually getting a histamine response that's bad. That's like
that's not bad. Histamine overreaction is bad. And so like I have swelling and itching that's going on for multiple days because of this yellow jacket toxin. But It's actually a normal response. This is and this is why the allergic response to bug bites, to bee stings, etc., is such a big deal, is because of that histamine response.
Right.
And if you get a normal histamine response to just a cut or a pinprick or whatever else, it itches a little bit and then it goes away.
I see.
Relatively quickly. But the itching is basically telling your white blood cells and your leukocytes within the body, come here and attack things.
Mm-hmm.
The histamine is part of telling your immune system that this is a site that needs to be attended to. When you have things that have more of an allergic response to them, that's when we call histamine response bad. Histamine is not bad. Histamine is part of the normal process. It's when histamine overreacts that it's a problem.
So you want a quicker histamine response that
Yeah.
improves healing is basically what you're saying.
Yeah.
Okay.
You want a quicker so that it peaks faster
interesting.
and then goes away more quickly.
Man, because I
for
always
healing.
feel like that's counter to the advice you're given, right? Like those of us who grew up in the walk it off generation was very much like, don't think about it.
Don't think about it. Don't think about it.
Yeah.
Yep.
But also,
But-
you would think about like the more I think about it, the itchier it's going to feel.
And that is true also. So we're not going to go into the details of the psychology of itch, which is which is actually, I think, related to this in a way, but it makes it more complex, right? Like
Mm-hmm.
I've been trying to distract myself from the itchy feelings on my hand all day long for the last 2.5 days, really. And this show's great because I'm talking and I might be itching occasionally. If you're watching the video, you've probably noticed me like itching my hand a few times already. But when you itch, what you do or when you scratch the itch, what you're doing is creating more subdermal or dermal issues that more histamines respond to.
Right.
So that's the issue. It's not scratching the itch. It's just what this study is showing, acknowledging the itch, acknowledging the injury, thinking about the injury, actually can be beneficial to healing, and that it makes it changes the way that your body responds to an offense, which I think is, instead of walk it off and run away from it, there is a certain aspect of attending to the sensations of it that makes a difference to your immune system coming and defending your body from whatever has happened. If that localized skin temperature increases enough, it will keep bacteria from getting in and creating an infection.
So this is really interesting because, stay with me here.
Yeah.
I have a toddler, and the current...
And they have higher temperatures than us anyway.
Well, that's true.
Great.
I was thinking more about like emotional health and how like the current wisdom related to how to deal with toddlers with their big emotions is very different from how it used to be, right? And instead of teaching them to like distracting them from the pain or the upset feelings, or telling them that it's not a big deal, or trying to ask them to calm down or any of these other things, instead you're supposed to let them embrace the emotion, recognize the emotion, work through the emotion, and then be done with it, right? And
how's that working for you?
It's working really well, I will say. Like,
good.
I am very impressed that like just like in the moment you're like, I can't believe I'm just gonna let this happen. But like you hold them, you tell them, everything's gonna be okay. I understand you're upset, like all that kind of stuff. And then when it's done, it's done. It has been resolved. Like, this, one of the things he says all the time now after he's been crying is like, I'm okay now. It's like, whoa, you just regulated yourself. That's the coolest. But like, that's very similar to this, right? Like you recognize the sensation, you recognize the injury, you attack the injury is now done, as opposed to like ignoring it and putting it off. I mean, and then it comes back later, right? Like it's kind of a similar theory.
My son was stung by a hornet's nest very like he was throwing rocks at a hillside, and then the hornets came out. They came and attacked him.
Oh my
And
goodness.
at the time, we acknowledged, we were like, that is scary. That's
Yeah.
bad. Like, you feel bad. Oh my gosh, of course you're crying. Do you hurt? Yes, you hurt. That doesn't feel good. Well, let's try and make it feel better. Just let's go through it. And he's he now wants to burn down our house because he knows that there's yellow jackets that close to our front door.
Well,
Ah.
two things can be true, you know?
Yes, I know. He's like, fire, Mom. We can burn it down. And I'm like,
oh
it's
my
right next to the
goodness.
house. He goes, we'll wet the house down. Everything will be great. Fire, Mom.
No, I don't think so. I think also you're just going to get some really angry yellow jackets out of that.
Angry, flaming yellow jackets. You don't want little flying insects covered in fire.
Yeah,
no.
that sounds bad. But yeah, that's cool.
Anyway.
I like it. Very
Yeah, so
interesting.
it's we just haven't had enough people talk about these things, and we know from anecdotal experience, even maybe our own experiences, of how focusing on things or thinking about things in different, putting our minds in different mindsets for healing, change things. And so this is like an actual physiological experiment showing evidence that voluntarily attending to a site of injury actually leads to a more regulated, faster immune response. That's better, that the outcome is like faster and better. And so this is it's interesting. So yeah, maybe we won't just ignore these things forever moving forward. Yeah, suck it up. Buck up, camper. Last couple of stories before we end for the evening. Sugar babies. Apparently, this one's fun. Once upon a time, during the Depression, not the Depression, excuse me. This was a study in England, Britain, between 1951 and 1956. People who were born in Britain, 64,000 people during this period of time were raised in a sugar shortage. So rationing of sugar was taking place because we're talking about a World War generation. The researchers studying these 64,000 individuals who spent their first 1,000 days, or a large portion of it, under sugar rationing, have a different metabolic holotype than people who were born after the sugar rationing. Although everyone after the sugar rationing ended in 1956 was eating more sugar.
Mm-hmm.
What they're saying is that individuals during their first 1,000 days who experienced longer periods of sugar rationing during that first couple of years of life, now as older adults, have lower incidences of breast, prostate, liver, rectal, and lung cancer.
Okay.
Additionally, they found that biological aging was slower for these individuals. So the individuals who underwent sugar rationing during the early stages of their life, they're biologically, metabolically younger than individuals who were maybe born later than them and did not undergo sugar rationing during that first early period of their life.
Mm-hmm.
Individuals that had longer periods of sugar rationing during their first 1,000 days had longer telomeres. So that it contributes to lower biological age and slowed aging and better general metabolism as you get older. And they found that there's a protein called granzyme B that gets higher the older you get with the immune system. It's a sign of slower aging at a cellular level. So that granzyme B was lower in these individuals who had less sugar when they were babies.
Well,
So
the first year, well, at least the first 6 months, there's no sugar anyway. It's kind of scrapped data. It's milk.
not necessarily. I mean, as soon as kids are able to start sucking on things that aren't a teat, you end up having applesauce packs. You have
Yeah, but that's after 6 months, right? So like the first 6 months. But
right.
also,
So
I think back then, I think people were breastfeeding way longer.
they were, but at yeah, there are very different,
So
very
yeah.
different things. But if the mother
Yeah.
also was eating more sugar,
Oh,
if the
interesting
family's
point.
undergoing
Oh, yes,
sugar
yes, yes, yes,
rationing,
yes.
so the
Yes.
nutrients coming to the breastfed are going to be different. So there are a lot of factors involved in this, and the researchers they really want to say it's not, that we're saying sugar is terrible or bad, whatever. This is just an interesting characterization that they have been able to pull out of the data that people who did not have sugar during their first 1,000 days have better health in later life.
Right.
And so it's kind of like, but what happened in between, that doesn't matter as much. I mean, it matters,
Right.
but this is the whole the body remembers the score. And so maybe you're setting the body up to, maybe people don't eat as much sugar because they weren't raised to have a sweet tooth.
Right.
Sure.
Sure.
There's so many questions that are still involved in this, but
yeah, it's also though like a generational snapshot. So there's like a lot of other potential variables.
I mean, now
involved.
everybody's, buying groceries at the dollar store. So,
Yeah.
America's
Yeah,
screwed.
sugar's in everything now.
It's in everything. I have to work to find bread that doesn't have sugar in it.
Yeah.
You have to work to find packaged products that do not include sugar. And the fact that that is not the first, that's not the basic state of all of your pasta sauces and everything. Like, what? Sugar, and it's the first ingredient in everything? Not my fault. This needs to be changed.
Yeah.
All right, final story.
Okay. Yeah.
Final story. Sometimes I wonder and worry about, I'm not moving enough. I have a desk job a lot. I'm sitting here not doing enough movement or activity. Would it be better if I got a job that was more active? Like, should I do construction? I'm not going to go do construction. My dad did construction. I saw what was in that. I'm not going to go do it. But researchers publishing at USC this week have shown that it's not just any type of physical activity that lowers the risk of dementia. It is the kind of activity that you're doing. So if you're doing physical activity as part of your job, it's actually linked to a higher risk of developing dementia. If you're doing activities moving for leisure time, that's the movement that is linked with lower dementia risk.
Interesting. Well, I mean, I know if you're doing movement as part of your job, a lot of the time it's repetitive movement. And so that's also going to cause physical ailments.
Wear and tear and strain. Yep.
But I think that's interesting that movements for work are related to dementia risk. That's crazy.
Yep. They found that the apparent harms associated with occupational activity are unlikely to be caused by movement itself. They're more likely to involve psychosocial stress, less job control, hazardous working conditions, and greater exposure to air and noise pollution. And independently, all these factors have been linked to dementia risk. So people in these jobs, because they're working and moving their bodies all the time, they may not have that time or energy for relaxing movement activities. So maybe you go out fishing or hunting or whatever, but are you running and I don't know. I don't think running marathons is good for you either.
But... No, that's like pushing your body to the limit. I I used to kickbox and I loved that. I still
I
do it
love
occasionally.
boxing.
Just because
Yeah.
it's very varied movements. It's a social activity. It's, at the beginning we do it to music. So there's all sorts of different kind of pieces of moving your body in rhythm and then doing different kind of dynamic movements that are actually related to things that you might do with your body at another time, right? Because that's also another thing is like, are your workouts something that you could actually use?
It's called functional workouts.
Versus vanity or whatever it's called.
Yeah.
Yeah. Mm-hmm.
I know. All I know is I just need to be able to get down on the floor, get off the floor.
Yes.
Can I lift things up? Can I put them down? Yeah. Can I turn? Here we go.
Yes, I look forward to re-engaging in physical fitness, hopefully soon.
Oh, life comes and goes with it.
Yeah.
Yes. We all need to set a timer once an hour to just like get out of our chairs and do squat jumps. 10 squat jumps. Just go.
Somebody walks by my office at work. They're like, "What are you?"
Kiki told me to jump, so I'm jumping.
Burpees?
Yeah. Gotta keep that cardiovascular health going. Come on.
I was talking to someone in the hallway the other day at work, and we decided to do a wall sit while we were talking.
That's awesome. That's great.
Anyway.
I love doing that stuff. I don't have anyone to do wall sits with anymore, so I just sit on the floor and pet my cat.
Yeah, that's good.
Eric Knapp says, sounds like life is bad for your health. And yes, of course, life is bad for your health, but it's understanding what situations are better or worse so that we can ameliorate those risks, right? Try and make workplaces safer, less stressful, better. I mean, if you're going to spend your whole life doing a rough job, hopefully we can make it safer and better so that you can live older better. I don't know. I think everybody deserves the right to age healthily, and we should be working toward that. So understanding what leads to unhealthy aging across the board is essential. Oh, wait, I probably just said something that would get like language filtered by NSF AI filters, didn't I? Anyway.
It's so crazy how politicized it is that we just want people to be able to like live fulfilling, healthy lives.
Can't we? Oh my gosh. That's all we want. I mean, some people want to live forever, and I don't want to live forever. I don't think that would be cool. I think it would be really weird. And I think the psychological aspects of living way past your, best by date would be weird.
I mean, I don't want to be a husk that can't communicate. That doesn't sound fun, but I would like to live forever.
Right. I mean, there are balances. Yeah. We're all. I want us all to live.
I understand
live
it's
long
not for everyone. I get it.
Yeah.
I want it.
I think no matter what, it's live long and prosper.
Yeah.
It's not a bad thing to think ethically about. But anyway, that brings us to the end of the show tonight. Have we done it?
We've done it.
Woohoo! I'm going to have a baby and a sugar ration. Hey now, hey now, hey now. No, I'm not. I don't know where that song
Kiki,
came from.
what?
I just made up a song. I'm making up songs now. I think we should come up with old 60s ditty bops related to modern science. Okay. Thank you all for being here for the show. Everyone in the chat room, thank you for being here. For your push-ups, your squats, your wall sits, and your comments. Thank you for joining us for This Week in Science. It's so wonderful to have you here with us when you're able to join us. It's just a magical moment to be able to share all these thoughts together. Thank you for listening wherever you are. Thank you to everyone for keeping our chat rooms great places to be. I mean, we cannot do it without you. Arnlore, Gord, everyone who's in the chat rooms maintaining compassion
Decorum.
and respect. Like, come on, everybody. Fada, thank you for all of your help with social media and show notes. Rachel, thank you for editing the show. And Identity 4, thank you for recording the show. And of course, thank you to our Patreon sponsors. Thank you to Aaron Anathema, Adam Mishkan, Alan Viola, Ali Coffin, Andrew Swanson, Arthur Kepler, Bob Calder, Brendan Minish, Brain Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Botts, Daryl Myshak, David Wilson, Dave Wilkinson, David E. Youngblood, Derek Schmidt, Don Mundus, E.O., Eden Mundell, Eric Knapp, Flying Out, Fred S104, G. Burton Lattimore, Greg Briggs, Jack Jason Olds, John Atwood, John McKee, John Ratnaswamy, Johnny Gridley, Ken Hayes, Kent Northcote, Kevin Parachan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessenflow, Merrick Jenkins, Noodles, Patrick Pecoraro, Paul D. Disney, Paul Ronovich, Pierre Velazarb, Robert Norland, Robert W. Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean Clarence Lamb, Shu Brew, Stephanie Embry, Stephen Alberon, Steve Leisman, aka Zima, Sue Doster, Teresa Smith, Tony Steele, Vegard Chefstad, and What the If? Thank you for all of your support on Patreon. I apologize for my pronunciation tonight. TWIS will be back next week.
At 8 p.m. Pacific time on Wednesday, broadcasting live from Twitch, YouTube, and Facebook channels.
Oh, yeah. And if you want to listen to us as a podcast, make sure that you search for This Week in Science wherever podcasts are found. If you enjoy the show, get your friend to listen too. Come on.
For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twis.org. And you can also sign up for our newsletter.
Oh, the newsletter. We love your feedback. If there's a topic you want us to address or cover, or a suggestion for an interview, please let us know on one of our social media accounts or send us an email. Make sure you put TWIS in the subject line so your email does not get spam filtered into
A fish shaped like a seahorse. A sea muppet, if you will.
Down by the sea. Yes.
We look forward to discussing science with you again next week. And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.